Hey everybody. So we're going to continue our lectures on the concept of prodrugs and drug delivery systems. As you remember, we're kind of entering this bio precursor prodrug section, which are a specific type of prodrugs that are usually activated by reaction like oxidation or reduction. Remember that when I distribute these PDF files on the iLearn site, I've been kind of building this sort of as I've been going along, so make sure you get the most recent PDF files for your records. Like today, I am updating this section of bioprecursor prodrugs and also adding more into the hydrogel section. The antibody drug conjugate section will be covered next week. All right. So what's a carrier-linked tripartite prodrug? Let's go back to that real quick. This just shows an example of it. This is in the last lecture. But 5-fluorouracil is a anti-tumor drug, sort of a nucleotide analog or nucleotide precursor, but it has the fluorine that is cytotoxic, and so this gets incorporated into DNA chains, and it sort of terminates the growth and kind of it's misincorporated into a growing DNA chain, and then this causes a lot of problems and cytotoxicity. And as a prodrug, the idea was that it could be connected to sort of a peptidal backbone, remembering that bacteria and fungi have enzymes called permeases, and they sort of non-selectively just uptake peptide-looking things, like this with this alanine on there. And once inside the bacteria or fungi, a peptidase cleaves off the alanine, and then the fluorouracil is expelled, and it's still within the cell. And this is super cytotoxic to bacteria and fungi, just like it's cytotoxic to human or mammalian cancer cells. Okay, so yeah, that was kind of a neat prodrug strategy, and it's called a tripartite because there's sort of an intermediate linker fragment. So the prodrug consists of the amino acid, and then this sort of linker fragment. And once inside the cell, the peptidase cleaves off the amino acid, and then this other fragment rapidly degrades, and that generates the 5-fluorouracil within the bacterial cell. Okay, so let's get into bioprecursors. So this is really just the nomenclature point of like different types of reactions that activate prodrugs. Because most of the ones, I guess all of the ones we talked about up here, the various carrier-linked bipartite prodrugs and this carrier-linked tripartite prodrug, these all use hydrolysis mechanisms, either ester or amide hydrolysis reactions, whereas the bioprecursor prodrugs usually use things like oxidation or reduction or some kind of other reaction to activate the prodrug. Okay, so the overview, similar in concept to the carrier-linked prodrug strategies, which use ester cleavage as an activation mechanism. Here we're going to use reactions like oxidation or reduction. So these might be P450 mediated reactions that we've talked about before when we discussed metabolism. So our first example is a unique activation method using protonation as kind of like a different reaction type, which is different than hydrolysis. We're just going to use acid as a reagent in your stomach, for example, to protonate a drug, and that causes its activation. It's kind of interesting. Okay, so this protonation mediated activation of a prodrug is exemplified by omeprazole, which is a proton pump inhibitor that we've talked about before in this class a few times, and it's activated by proton. Okay, So in the history of these proton pump inhibitors, a bunch of molecules were looked at kind of in the early to mid-70s for the inhibition of protein. It's the proton potassium ATPase, which is also called the proton pump. It secretes acid into the stomach, and that's used for digestive purposes to break down foods and things like that. And it also causes symptoms like heartburn and peptic ulcers and different things like that. And so it's useful to inhibit this protein, this channel protein, because it's a channel protein, in order to relieve some of these symptoms. Okay, so yeah, peptic ulcers, heartburn, GERD, which is gastroesophageal reflux disorder, H. pylori infections, which is a bacteria that you can get in your stomach, and that has links to cancer. And if you have an outbreak of H. pylori, it can cause other problems. All of these symptoms can be treated with proton pump inhibitors. So some of the compounds that are identified in the optimization process were shown here. They kind of vary from less complex to more complex, starting kind of with the thioamide pyridine linked compound. And then we have a dihydroimidazole with the pyridine. You can kind of see the resemblance of some of these structures as they kind of varied the structures. And this is, in the 70s, we kind of knew how to make molecules back then, but not very well. And we started learning more about synthetic chemistry kind of into the 80s and beyond. Then they went from the sort of dihydroimidazole type thing to a benzimidazole. So it actually added a benzene. And then rather than a sulfide, people started looking at sulfoxides, right? And then adding a methyl group and maybe a couple things on the benzimidazole, that had kind of an effect also. Let me check something. Okay, and later, in the kind of the most recent generation of these proton pump inhibitors, they added a couple more things like 2 methyls and the methoxy group on the pyridine, and that kind of enhances the nucleophilicity of the prodrug, of the molecule. And one second, sorry. And this will have an effect on the mechanism that we're going to see. Okay. And also they changed around the substituents on the benzimidazole, so they added a methoxy group there, and all of this kind of affects the electronics of the drug. Okay. All right. Let me see. I'm researching one thing. I'll just take a second. Sorry. Yeah. Okay. And so omeprazole is pretty much one of the modern kind of standard proton pump inhibitors. It's a generic drug. A lot of companies sell it as omeprazole. It's a multi-billion-dollar drug and it was originally patented by, I think it was AstraZeneca, and when the patent ran out, they started looking at ways to maybe make this profitable again, and I believe so. It turns out that these molecules have sulfur chirality, looking at the molecule, it doesn't look chiral, but remember that sulfoxides are chiral, so the oxygen can be kind of coming towards us or going away from us. And that's the case for any of these sulfoxides. And anyway, later AstraZeneca, I believe, created S-omeprazole, which is the S chiral version of this instead of R, and it's efficacious also, but there's some question whether it's just a method to get more money out of the drug by having something else they can patent that basically does the same thing. And generally speaking, omeprazole is very efficacious. It's one of those things on the World Health Organization essential drugs list. So it does its job really well. And the question of whether the sulfur chiral center helps efficacy is, I think, a little unresolved and maybe controversial. Okay. All right. So, like we said, the proton pump inhibitor is activated by proton. So the proton is part of the activation reaction. I'm going to show a mechanism for this. These are a couple good papers down here. Jack's 2004, page 7800. And also current gastroenterology reports, 2008, page 528. So this kind of outlines some of the mechanism I'll be showing. So it's activated by proton. It irreversibly binds to cysteines in the channel protein. So it actually attaches to proteins. And we've talked about these sort of irreversible inhibitors before. And that if it's selective, it could be okay. I mean, you always have to worry about a drug that's kind of permanently modifying your proteins, or certainly DNA would be more of a problem. It turns out, though, that the protein's kind of recycled by our body. So even if you're permanently deactivating it, it doesn't mean that it's gone forever, because our body's able to synthesize more of it. That also means, though, that the drug can actually have a effect for maybe a couple days. If the drug kind of permanently deactivates the protein and it takes maybe a couple days to resynthesize the protein, the drug can have an efficacious effect for a while. Okay, so this is kind of how it works. And we talked about how this is omeprazole, how there's a couple methyls there on the pyridine, and also the methoxy, and all of these are kind of electron donating substituents. What happens is the nitrogen of the pyridine attacks the benzimidazole C double bond N, and the double bond N grabs the proton. So that's how it's kind of absorbing protons in this process. It's activated by a proton, and it's also it's going to inhibit the proton pump, right? But that's through kind of a different mechanism, because it actually obstructs the proton pump. It binds inside the channel. We'll see how that might happen in a crystal structure in a second. Okay, so when this happens and this attacks, the nitrogen attacks, the double bond grabs a proton. Now it sort of cyclizes and forms this, looks like a 5-member ring, and then the nitrogen swings back and breaks off this, I think it's called a sulfinic acid, S double bond OH, and then the other benzimidazole nitrogen swings back in and forms another bond, and so now we have this 6-member ring, and then cysteines in the active site of the proton pump will attack sulfur and form a disulfide linkage like this. And when this happens, now this is permanently attached to your proton potassium ATPase molecule, protein, your proton pump. Okay, and so for omeprazole, it's binding to specific cysteines that have been characterized, cysteine 813 and cysteine 892. Other proton pump inhibitors bind to different cysteines, like pantoprazole and lansoprazole, bind to cysteine 813, which is the same, but also cysteine 321. And so the drugs kind of have there's different effects on turnover of the protein when the cysteines are attached to the drug like this, and it kind of depends on which cysteine it is attached to. So there's these kind of extra concerns about which cysteines the drug's attached to, and how that affects like the turnover and recycling of the channel protein. Okay. All right, so back to omeprazole. There's no X-ray crystal structure that I could find that shows how it's actually attached to these cysteines covalently. It would be kind of fun to see. I always like to see protein structures if we can. It is concerning maybe that omeprazole and the related proton pump inhibitors seem to covalently and irreversibly bind to this protein. Does the protein recover? And like we said, yes, it does seem to. You can imagine it just be resynthesized, and your body's constantly making proteins. It does not appear to be a big problem since the protein is continuously resynthesized or turned over. And also the drug can have an effect for a couple days, which is probably not a bad thing. I mean, if you're suffering from some of these ulcer problems, this is a kind of a miracle drug, so it may be actually beneficial that you're not generating large amounts of acid in your stomach. You could also imagine that the covalently attached intermediate, the disulfide, could break down with nucleophiles, maybe glutathione, which we know is a nucleophilic thiol, and you can imagine that it might attack the left sulfur or the right sulfur to kind of cleave the bond. If it attacks the left sulfur, then that regenerates the cysteine. And glutathione, being a nucleophile that's kind of present in a lot of tissue types, could theoretically do this. So there's ways the disulfide bond could be cleaved. This is also discussed in these two papers. Nevertheless, a company discovered a reversible PPI, so one that does not bind to cysteines, and it has a lot of efficacy, and it's maybe a little better for the H. pylori infections. There's other reasons that this is useful as a alternate proton pump inhibitor drug. And deals with maybe some of the side effects that omeprazole have and the other sulfoxides. And it does not irreversibly modify the proton pump. And it also has an X-ray crystal structure. So again, anytime we have X-ray crystal structures, it's always kind of cool. So this is going to be an X-ray crystal structure of the potassium proton ATPase proton pump bound to the reversible, so non-covalently bound inhibitor. And the inhibitor is called vonoprazin. I'm probably mispronouncing that, or vonoprazin or something. So this is pretty cool. This shows the structure of the proton pump. Up top is the cytoplasm of the parietal cell. There's a membrane that separates the parietal cell from the gastric lumen, which is sort of the stomach lining inside the stomach. Okay. What happens is protons are pumped from the parietal cell outward into the gastric space, and potassium is pumped inward. So it's like an anti-Porter type channel protein. There's the drug sitting right there in the assembly of all the alpha helices. It's called vonoprazin. These are two papers actually. Digestive. Let me see. I'm gonna have to double check these. Oh no, this is right. This is right. Yeah, Yang A. Digestive Disease Science is one paper, and then Abe or Abe Nature 2018. So this talks about this drug quite a bit and some of its efficacy. And the Nature paper is specifically on the X-ray crystal structure. And usually if you have a pretty cool crystal structure that's never been solved before, there's a lot of discussion on the structure of the protein, and that's all in this Nature paper. So it's a cool read as you're learning more about structural biology. Here's a zoom in of just a different perspective of kind of where the drug is sitting right there. And you see some of the elements, and there's a sulfonamide, S double bond O, fluorophenyl, and then the N-methyl group on it's kind of hanging out underneath there. The structure looks a little funny because it looks like there's two little bonds to red and two little bonds to red from the sulfur. Well, of course, that's just the double bond because those are double bonds. So this is just the way the pi mole is drawing the sulfonamide. Okay. Another thing about this X-ray crystal structure, it binds near those cysteines, like cysteine 813 and cysteine 892, which does not do it covalently. It's a reversible inhibitor. So the protein, or the small molecule, has an affinity for this channel protein. It binds in there, but it doesn't have those weird mechanisms that cause it to attach, I guess. If you look at the structure again, this is a sulfonamide. It's S double bond O, double bond O. Omeprazole and those other inhibitors, those are all sulfoxides, and that kind of allowed that sort of proton activation process and then the covalent attachment. So this works a little differently. It just reversibly binds to the channel protein like this. Okay. So this just shows the same perspective, well, slightly different perspective of how the drug sits, and it shows where those cysteines are that would actually interact with omeprazole, for example. So the cysteines actually bind to omeprazole and permanently attach. So it's near them, it just doesn't bind to them covalently. Okay, so now let's get back into bioprecursor prodrugs and other mechanisms like oxidation and reduction. That was a pretty cool example of how protonation activates omeprazole as a proton pump inhibitor, and how it binds to cysteine. Okay, so this was an interesting example to me. It's P450 oxidative demethylation to generate benzodiazepam anti-anxiety drugs like Xanax and Halcyon. So this actually happens in the body. I don't know if it's actually clinically used, but it is an interesting kind of biological reaction process that happens. So if you look at these molecules, these are examples of benzodiazepines, which are kind of like Valium and other related molecules, and these molecules are called Xanax and Halcyon. Anyway, so what happens is P450 does an oxidative demethylation of one of these N-methyl groups, and actually does it twice. So you can see there's two methyls, now there's one methyl. And then after it does it the second time, you have an NH2, and that reacts with a ketone. And so then it cyclizes to make this hemi-MnL intermediate. And then the nitrogen swings in, kicks off the hydroxy, and now you got a more like an imine type derivative. And these are the benzodiazepines, which are a couple of them. There's a whole bunch of them, maybe like 1,000 benzodiazepines, and a lot of them are used clinically as sort of anti-anxiety drugs. So that's Xanax. That's one of them that's used. Alprazolam, and then Halcyon, triazolam, and these are kind of abused substances also, like Valium is also. And what about P450 reductive cleavage of things? And the only examples I'm going to give are for azo linkers that are a prodrug strategy in the early sulfa drugs. So these are, remember the PABA analogs, that are used to inhibit dihydrofolate reductase. It's one of the antibiotic types, drug types. Okay, so first one is called Prontosil. It's a sulfa drug, PABA analog, dihydrofolate reductase inhibitor, and it's one of the, like, historically, it's one of the earliest examples of a prodrug. And it's also one of the earliest discovered antimicrobial drugs. Okay, so the idea is that here's an azo linkage. This would be reduced, and I'm imagining it's P450 NADPH. I'm not 100% sure about that. That generates the PABA analog. This is called sulfanylamide. It's a sulfa antibiotic, and then triaminobenzene. This was, I think, discovered like in the 30s or something like that. So here's some historical images of what the pharmaceutical packaging used to look like. There's another one called sulfasalazine, which also had the name MNB 693. So this is a slightly more complicated molecule. It was also kind of in the 40s probably. So it had a pyridine on the left, sulfonamide linkage, and then a azo linkage, carboxylic acid and hydroxy. And this would also be reduced, I guess, again, and I'm not 100% sure, but you need to do a reduction. That's why I would imagine there's NADPH in there. But that cleaves the azo linkage and makes an amine, two amines, and this creates sulfapyridine and 4-aminosalicylic acid. And this is also considered a PABA analog, so it should probably have the same kind of effect as sulfanylamide. Is also kind of historical, and here's the historical drug packaging. You can see the date of supply is December 1943. This one actually has another historical link. It was a drug that possibly saved Winston Churchill's life, because in World War II he had pneumonia, bacterial pneumonia, and this is the drug he was given, and it saved him. And there's a famous quote, this admirable MNB, referring to MNB 693, from which I did not suffer any inconvenience, I guess, maybe he meant side reactions, was used at the earliest moment, and after a week's fever the intruders were repulsed. I guess by intruders, I don't know, maybe he meant the bacteria, the infection. So this is Winston Churchill, 1943, in a wartime radio address after being treated for bacterial pneumonia. Okay, so that's it for bioprecursor prodrugs. There's not as many examples as the carrier-linked prodrugs that we showed with esters. There are more, but I just wanted to give you guys a taste of this. Okay. So let's get into hydrogels as a vehicle for drug delivery now. Okay, so let's talk about what a hydrogel is and some of the methods that these can be used to deliver drugs. First of all, so we need to talk about how drugs are linked to maybe or conjugated to like a protein or a macromolecule. And I realized that you may have not learned this before, so let's first talk about ethylene glycol, which is called 1,2-ethane diol. You've probably heard of this substance before. It's a pretty simple molecule. It's a 1,2-dialcohol. It's used as a synthetic reagent. It's used as an antifreeze in car radiators and things like that. You've probably seen it before in terms of protecting group chemistry and organic chemistry. So this sort of dihydroxy group reacts with ketone and makes a protected ketal or acetal, and then you can do additional reactions on this. It's also used as an antifreeze. It's actually toxic. It's not really used as a solvent, or like, well, it's used as a solvent, but like in pharmacology or drug formulation, it's actually kind of bad because there's some reactions that happen metabolically. I think it oxidizes to oxalic acid, which is the carboxylic acid on one side, carboxylic acid on the other side, and that has some toxicity issues. As a antifreeze, it's actually, well, it's kind of a sweet-tasting substance, so it has a sweet flavor, and that causes a lot of poisonings. So people might accidentally poison their dog or their kids or something because it has sort of a sweet flavor, and people take it unintentionally. So what's polyethylene glycol? So polyethylene glycol is kind of a polymer version of ethylene glycol. This is not toxic, and it's actually used for a lot of things in drug formulations, and also to maybe link a drug and some other thing, maybe an antibody or other macromolecule. It can be attached to PEG. So PEG, you'll hear about PEG a lot in drug discovery and drug delivery. So it's a polyether compound. It's less toxic. It doesn't have that kind of, you don't have to worry about it forming oxalic acid. It's biocompatible. It's aqueous soluble, so you can have long chains of these, and it's kind of like a long chain aqueous soluble polymer. It's commonly used in medicine and material research, and there's a lot of uses of PEG. It's often used as a linker between small molecule drug components, maybe antibody drug conjugates, which we'll talk about later in this course, etc. Okay, so being a polymer, there's different sizes of it, and you'll see these occasionally. It'll be like PEG 400, PEG 3350, PEG 1000. What does the number refer to? It's the molecular weight in kilodaltons. PEG 400, 3350, 1000, or like different sizes of PEG. There's also different branched or functionalized forms of PEG that can be attached maybe to a drug molecule, and that's going to be useful in this hydrogel discussion that we're talking about. So you can imagine like a carbon in the middle and having PEGs kind of emanating outward. You could imagine doing the same thing but with amines. So these are like nucleophilic terminated PEG-like structures. So it's, and the N can be a big number, so these can really emanate out kind of over a long distance. There's also, in addition to these sort of nucleophilic ones, and nitrogen being more nucleophilic than oxygen, you can imagine like sort of a carboxylic acid terminated PEG, and then this thing is a leaving group. It's kind of like a chloride or something. It's a well-behaved leaving group. It's maybe biocompatible. Okay, so a chlorine or other thing can be attached as a sorry, I'm trying to say this kind of resembles like the idea of a chlorine, right? It's called succinimide, and succinimide is a well-behaved leaving group. So nucleophiles can kick it off, okay, and attach to it. All right, so you can envision that these sort of branched PEG-looking things could be interconnected or cross-linked into sort of an immobilized, maybe it's aqueous. It's not soluble, but it does dissolve water, and water can be surrounding it, and it forms sort of a gelatiny mass, polymeric mass, and it can be used as a drug delivery vehicle because you could attach maybe a drug to one of these arms or something, and then through the proper cleavage conditions, the drug can fall off, and then treat your tumor or whatever. So this is an example of a hydrogel. So a big issue then is if we have a drug or something attached, and assuming this is kind of all cross-linked and there's a lot of these together and they form little masses of these branched PEGs. Assuming you have a drug on there, like how can you release the drug? Because if it's permanently and covalently attached, well then that's not going to help. You need to be able to slowly and controllably release the drug molecule. And we'll talk about some of those mechanisms shortly. Okay, so let's show examples of drug release strategies using hydrogels. So this is actually kind of early literature, 2008, from a journal called Polymer, and it showed like if you have like a hydrogel matrix, there might be negatively charged particles on the hydrogel, and then your drug might be positively charged. And so there's an ionic interaction, and the two things interact, and then the drug kind of gets slowly leached out, falls off, and does its drug thing. It has this drug effect. And yeah, it's kind of an ionic interaction. Another idea is maybe you have the polymer backing, and then there's some kind of cross-linker, a cleavable cross-linker, and it could be cleaved by an enzyme, so maybe a peptidase or protease or something, or other methods to cleave the linker, and that would also release the drug. So these are kind of ways you might address that. A lot of labs and companies have been investigating alternate and additional methods to cleave drug molecules from a hydrogel to facilitate drug delivery. And I'm going to focus on a company called ProLynx, which is in San Francisco. I think it's maybe China Basin or Mission Bay. It's kind of, it's by UCSF Mission Bay. And they're a company that's based on using technology like this and like special drug cleavage methodology to release drugs that can be used, to treat a disease like cancer or something. Okay, so beta elimination can be used as a cleavage mechanism to release drug from a hydrogel. Let's review some examples to review some of our organic chemistry knowledge of beta elimination. You've probably forgotten all of this, but let's talk about it. So, as you learned early in your organic chemistry education, bases can take protons off a molecule and then sort of in this concerted process cleave off a leaving group. And then that generates an alkene, right? And you get a BH and an Lg minus, some kind of bromine or chlorine or some kind of leaving group gets kicked off, and you get an alkene. What is this an example of? Which reaction mechanism is this? This is an E2 reaction. And usually you need a strong base, so maybe alkoxides or those are used. DBU is a base that's sometimes used synthetically to carry out E2 reactions. What does the 2 mean? The 2 means that it's biomolecular, so you have a base. Both of these are in the rate-limiting step, the base and the substrate. Okay, substrate can be primary, secondary, tertiary. So this is a primary substrate. Okay, so what happens if the leaving group first falls off to make a carbocation, and then some kind of weak base will take off that proton and generate an alkene? This is not an E2 reaction. 2 being, again, bimolecular. This is not a bimolecular reaction. This is a unimolecular reaction because the rate-limiting step is the loss of the leaving group. This is a slow step, right? The leaving group usually doesn't want to spontaneously fall off. But once you form the carbocation, the base, in this case water, will just take off that proton and make the alkene. So this is an E1 reaction. So it's a weak base or solvent. We call this often solvolysis, or when you learn solvolysis, we usually teach SN1 reaction, which is kind of like this, but leaving group goes away, and then a water or alcohol nucleophile attacks the carbocation. Okay, so E1 often accompanies an SN1 reaction. Okay, so now this is more relevant to the cleavage method that ProLynx uses. What if you have a base and you have a proton and a leaving group, but what happens is, and maybe the leaving group is not as good, like as a chlorine or bromine, but it is the leaving group. What if the base takes the proton and leaves you with a dot negative, charge, and then this is semi-stabilized. So the negative dot negative is semi-stabilized by the sulfur, the double bond O, and maybe R groups out here that may affect the stability, right? Maybe if it's an electron withdrawing group, if there's an electron withdrawing group, it's pulling electrons, well, that might suck the negative charge kind of inward and create a stabilized complementary interaction between the R group and the negative charge. Well, then this can hang around for a while, and then and then the negative charge can kick off the leaving group. Okay, and so this is a another cleavage mechanism. It's not E2, it's not E1, but is it bimolecular or unimolecular? Like E2's bimolecular, E1's unimolecular. Well, it depends on which reaction is the fast reaction, and it turns out that the fast reaction is removal of the proton. The slow reaction is the kicking off of the leaving group. So the slow reaction kind of depends on just one substrate. So it's going to be a variation on E1, and the name is called E1CB. CB stands for conjugate base, right? Okay, so it's kind of like you have a fast reaction and a slow reaction, and since the slow reaction, the rate limiting reaction, is unimolecular, that's why it's E1CB. Okay, and then we're going to call this thing up here, in this case it's like a sulfone or sulfonamide or something. We're going to call it a modulator because this can be varied. Depending on the electronic properties of this modulator, it can affect the speed of this reaction because it'll affect the stability of the negative charge, right? Okay, so what we're going to do now is E1CB reaction applied to drug release. And so you can imagine now, let's step through this kind of slowly. You might have like a carrier hydrogel type thing, or by hydrogel I could mean like a branched peg or something like that. And there's going to be a linker, maybe in a drug molecule kind of hanging out here. Well, what if the base takes the proton? You have a temporarily stabilized carbanion intermediate, and then that kicks in, the negative charge kicks in and then kicks off the linker and the drug. And then the linker drug piece can degrade to actually generate the drug. This could be a drug delivery device, right? If you have this sort of like a big hydrogel thing, and then you have a modulator, and all you have to do is maybe vary the modulator to vary how fast the drug is cleaved. So this is one of the technologies that this company ProLynx kind of created, and they popularized as a cool method to deliver drugs. And there's another thing you could do is E1CB reaction applied to hydrogel degradation, hydrogel degradation. So in addition to just releasing the drug, you could imagine maybe having this reaction kind of cleave different components of a hydrogel. It's the same exact thing, it's just now I say hydrogel 1 and hydrogel 2, and the same kind of reaction occurs. When I say hydrogel 1 and 2, what I really mean are branched pegs. So it's not necessarily a hydrogel per se. It's like a branched peg 1, branched peg 2. Anyway, the base takes the proton, and you get their conjugate base. It's an E1CB reaction. And then this is stabilized for a while, and then the negative charge kicks in, kicks off the other piece, and now you've kind of cleaved it, right? And this is a method that can be used to kind of cleave two of these branched pegs. Okay. This is also something that ProLynx uses. Okay, so the rates of E1CB beta elimination can be fine-tuned by varying the modulator. That's something I just said. Here's the paper. It's a nice kind of early paper for this company. Hydrogel drug delivery system with predictable and tunable drug release and degradation rates. Well, that sums it up, right? Being able to modulate the drug release and degradation rates kind of by playing around with the modulator. And this one of the cool results that they published in 2013. Okay, so this again is the basic principle. Base takes proton, you get a stabilized conjugate base, it swings down, kicks off Y, then you got these two pieces. They looked at some mod pieces, modulators. These are a couple of them, and you can see that they vary quite a bit, and it's great because you can control the release rate between like kind of 1.5 days, and these are half-life. So by half-life, T1 half, we kind of mean that like after 1.5 days, half of it gets degraded, right? and then so you still have the other half. Anyway, so as we kind of transition from this chlorophenylsulfone to the phenylsulfone to the methoxysulfone, the cleavage rate is decreased, or the half-life increases, I guess. Yeah, methyl sulfone is 19 days, and this sort of what is that, morpholine derivative is about 30 days, and then a simple nitrile as a modulator extends the drug release to about 100 days. We'll see this one again, so keep this in the back of your mind. If you want a drug to be really gradually and slowly released, which one of these would you use? Maybe the nitrile one, right? Okay, so I'm just going to show two examples of sort of how ProLynx, this company, is developing drug delivery devices. This one is going to be sort of a PEG-based hydrogel. So it's polyethylene glycol. It's a hydrogel. You have some kind of linker and then a drug molecule type thing. So it's useful for extended release drug delivery. The hydrogels can be degraded very slowly, and so you could almost have a hydrogel that might last for months or a year. So you could inject this into your skin, or like a site of a tumor or some disease that you're treating, and then have the hydrogel kind of last for a long time. So it could last for weeks, upwards to a year in like a human or mouse or model organism, and slowly release the drug. Okay. Given the ability to fine-tune these modulators, ProLynx is able to fine-tune the release of the drug molecule and as well as the degradation of the hydrogel. So this is a different paper, recent. This is last year. International Journal of Polymer Science. I think I'll try to post all of these on the course website. This shows the hydrogel. This is kind of like the polymeric cross-linked hydrogel, and there's the little drugs in there. And then the idea is you'd fine-tune the modulators so that you could kind of slowly release the drug, and then some of the drugs are going to kind of come off with the polymer components, but generally speaking, you're going to have like free drug be released, and then the drug's kind of gone, and then you have even a slower gel degradation. So then the degradation kind of goes away also, and you form monomers. It's kind of critical that when you form these monomers, what do? Do they just kind of hang out? Do they just stay at the site of your tumor, or where you injected this drug? If it just stays around, that would be a problem, because that might cause toxicity, or you don't want to just have junk in your body that doesn't ever get excreted or eliminated, right? So fortunately, these monomers usually get kicked out and sort of dissolved away and excreted through your kidneys and urination. Okay. So then, the title of this figure in this paper, I shortened some of the figure legends a bit, but it's depiction of chemically controlled drug release and subsequent degradation of tetrapeg it says hydrogens. That's my spell checker. It should say hydrogels with beta eliminative crosslinks. Okay, so you're using this beta elimination cleavage method. Okay, the other cool thing about this is when we think about how we use this clinically, you generate a slurry of hydrogel particles, and they form little microspheres, little spherical particles, and they can be sucked into a syringe and injected. And so you can imagine like a mouse or a human, you basically create these little spheres, and they can be pulled into a hypodermic needle. That's important if you want to inject them. And it can't be like a huge needle, maybe it depends on the gauge. But if it's a really painful needle, well, that's a problem. But generally speaking, these things can be injected. So easily done at specific treatment site. So we might do intratumoral administration. So we inject into a tumor site, for example. That's an idea how you do this. This is a pretty cool graph from this paper that shows different variations of the modulator, and Also, we're going to show this alpha lysine and epsilon lysine on the next slide. These are like kind of different arrangements of how they put this all together. But the chlorophenyl sulfone and the phenylsulfone, methyl sulfone, and sorry, methylphenyl, 4-methylphenyl sulfone, methyl sulfone. All of these have different degradation rates. And what this is showing, it says fraction soluble, is that if you have these different polymers, these different beta eliminative drug conjugates, fraction soluble kind of means like the amount of the material that dissolves, that degrades. So what this is really doing is not really following the first step up here. It's more like the degradation of the hydrogel into the monomers. And you can see, for example, the red one, 4-chlorophenyl sulfone, you can see it kind of degrades and then it's completely dissolved, right? And TRG, I think, was the time of reverse gelation, I think is the name they used. Time of reverse gelation. I think they use that in this paper. And reverse gelation means dissolve. It's like you have a gel and we're reversing that, so we're ungelling it or whatever, right? And so each of these have these different TRG values, and they go upward to 75 days or whatever in this example. Okay, so based on the different substituents, you can get different TRG values. So from this paper, in vitro solubilization or degelation curves of tetrapeg hydrogels with cross-links of different cleavage rates. So it's really cool that they can fine-tune the rate of cleavage. And there's the other thing to note is in addition to the substituents, they also have these different lysine flavors, and we'll show that on the next slide, what these alpha lysine ones and epsilon lysine ones look like. So now this will just show a little bit of the synthesis of these and kind of how we're building these conjugates, or actually how ProLynx does this to generate cleavable hydrogels. Okay, and so what this is showing is sort of how the attachment process works. Early in this course, we talked a little bit about click chemistry, and what we're seeing here is let's go from left to right, I guess. PEG 20 kilodaltons. So this is a PEG, a branched PEG. That's what the 4 means here. It means there's 4 of these on the PEG, right, off the central PEG. I think the central atom, I think, is a carbon atom. And what click chemistry did, if you recall, is it snaps the azide, an azide functional group, onto an alkyne, and it creates this thing called a triazole. So these are little triazoles created by click chemistry. So the name is click chemistry of that. And what I'm going to go into a little depth on the next slide, is a little tangent on why do they use this fluorocyclooctane thing? That's kind of weird. I mean, if this sort of works for just, it's really just, alkynes and azides, why would you do this kind of weird one with the fluorine and the 8-member ring? Well, first of all, that's a weird strained alkyne. That kind of sums it up right there, right? Because an alkyne is a linear functional group. It's like carbon. It's 180 degrees. Carbon, usually. So this is strained. This is a very strained cyclooctane. And also the fluorine affects the electronics of this. What we'll see on the next slide is that this is it's a very strained alkyne, and so the reaction actually goes very spontaneously. So this alkyne reacts with one of the azides, and then forms the triazole pieces. So we were just talking in the last slide how there's a couple different lysine frameworks. I think they're calling this the So when this azide attacks, I think they're calling this the alpha lysine framework, because you're attaching to they're using the alpha amine of the lysine to have these two pieces held together. So here's one peg, branched peg, here's the other branched peg, and we're using the alpha amine of this lysine. The other thing they're not really showing in this slide is that they're attaching like a drug at the R position. It's kind of like you have a peg piece for one, you know, this is building the hydrogel, so you have one peg piece, you have another peg piece. It's all kind of attached with a cleavable linker. This is the cleavable linker, right, with the modulator, right? And and then the R group is doing the same exact thing. They're just not showing it because the slide gets complicated. But the R group is a potential drug that can also be cleaved. The R group can be cleaved as a drug release strategy. And the difference between the top and the bottom here is that here we're linking on the alpha amine of the lysine up top, and here we're linking on the epsilon lysine, epsilon nitrogen of lysine. So if it's alpha, beta, gamma, delta, epsilon, this is called the epsilon amine of the lysine. Overall, though, it looks kind of crazy, but it's really just you're just using the click reaction to snap things onto this alkyne, and then you have this crazy framework, but all that really matters is you have a cleavable method. So the cleavage, just, you know, the modulator, you can control the rate of cleavage. That proton can get torn off and then cleave the hydrogel. Okay, and so this is formation of tetrapeg hydrogels with cleavable crosslinks connected to the alpha or epsilon amine of the lysine residue. All right, so what I want to do is take a little bit of a detour out of drug delivery, just because I want to talk about this a little bit more. This is super cool. This is probably going to win the Nobel Prize at some point or another, because of all the cool things that this has created. And I wanted to do this at some point in the course, and I think this is the time to do it. So, quick detour into this click reaction. Oh yeah, this is what I was saying. R groups can show another cleavable linker tethered to a drug. So you have this same kind of cleavable linker framework, maybe with a different modulator up here at the R group. Okay, so mini-tangent bioorthogonal click reaction. This is crazy work by Carolyn Bertozzi. She was at UC Berkeley, now she's at Stanford. This paper is really cool. It's From Mechanism to Mouse, a tale of two bioorthogonal reactions. The journal is Accounts of Chemical Research, 2011. So what we're going to show is a couple of variations of the click reaction, and starting with kind of the original one. This just shows, like, you have an azide and an alkyne. You can cook them, and you get this sort of mixture of triazole products. And the azide can align up on one orientation or the other, so you get an isomeric mixture. Okay. And then there's a copper version, and this will usually give you one stereoisomer, and so the R group is on one side and the R prime is on the other, and it makes a triazole. What's really cool is these are great ways to link two pieces of a molecule, right? And what Carolyn Bertozzi really, I don't know if she discovered this, but she definitely, she maybe did discover it. She didn't discover these two things. These are really old. What she discovered is there a way we could do this without copper? Is there a way we could do this without copper? Because copper is kind of toxic. And if you want to do this maybe in a living organism, the copper will totally kill the organism, right? So let's try to do it without a copper. And so what she found, and this is what I just showed you, and that what ProLynx is using, is that you can have these cyclooctines. It's like a strained molecule with two sp hybridized carbons. So this is a very unhappy molecule, and that an azide can react with this in the same kind of click fashion at a room temperature without copper. So what's a good name for this? It's the copper-free click reaction. It's the copper-free click reaction, okay? All right, so copper-free click reaction. And so this is the one that, in variations of this, that are really used a lot for pretty much modern click chemistry. Some people still use copper mediated click if you don't need to put it into a human or something, or just a synthetic reaction. This is still a wonderful reaction too. But this is super cool, and it opened doors to a lot of neat research. So what Bertozzi did then is they looked at this, like, well, if this reaction happens, can we maybe enhance the rate of the reaction by changing substituents all over the place? And what this is showing is the K value is the rate. It's a rate constant, right? And so this is showing kind of a bad one. It's like, it's a little bit slow. The number's kind of small. And what they found is if they vary the things around the cyclooctane, that they can actually speed this up a bit. That's also kind of a bad one, right? But what they found is they give them fun names too. This is oct, and this is alo, and this one is called mofo, which is a higher rate reaction. Just by adding the fluorine, really, and maybe changing the framework a little bit, they get a pretty sizable jump in the rate. It goes 4 times faster, right? 2 times faster than oct, and 4 times almost greater than alo. And dibo is another one where they have 2 benzenes on both sides. And this is a pretty sizable rate increase from where they started with. Then they had something called difo, which is even better, and they had 2 fluorines, and then they can kind of link things onto the terminal carboxylic acid out here. And then they also found something called barak, which is even faster. If you look at the rate constant, it's much greater than all of these guys. I think the Bertozzi lab did the most work with the difo one when they discovered it. And I'm going to show a couple of really cool examples of that real quick. Okay, so then they here's a couple of results from another paper they did in the Proceedings of the National Academy of Sciences. Copper-free click chemistry for dynamic in vivo imaging. Okay. In vivo imaging is kind of doing imaging of a living organism, which is pretty crazy. Think about this a bit. If we have a cell, and we can grow the cell with these, maybe a sugar with an azide on it. Oh, azides are the things that react with click. So you can grow, and azides happen to be kind of bioorthogonal, meaning they don't kill the organism. The azide's actually kind of biologically happy functional group. So you can grow the cell with this azide labeled sugar, and sugars often are used on the surface of the cell for signaling processes. Now we have a cell with azides on it, and then the idea is we could use the difo maybe with a fluorescent label on it, and now we can label cells with maybe different color dyes, and maybe at different stages of like a development or a cellular development or something. So this shows the difo species, and this just shows 2 examples of fluorescent dyes. These are Alexafluor 488 and Alexafluor 568, which are different colors. So these organic dyes have different colors. This one's kind of blue, this one's kind of yellowish, right? And then you can do really neat studies where we're looking at maybe the transformation of cells and the amount of the sugar and where the sugar is being localized on the cell at different points of a development or something. And I'll show a couple of slides of that. I know we've deviated a bit from the drug discovery or drug delivery talk, but I'm just trying to show how cool this is of using these cyclooctanes. Okay, this is the second slide of the mini tangent using the Bertozzi copper-free click reaction. So this is in vivo imaging of zebrafish embryonic development, Bertozzi, and she's using the paper. This is another really good paper, visualizing the enveloping layer glycans during zebrafish early embryogenesis, which is like the early embryonic development of a zebrafish. Okay, so this is the same idea that I just showed you. You have the cell, they incubate it, a living cell, incubate it with these azide-containing sugars, and then they can use these really cool dipho fluorescently labeled dyes, and they do a copper-free click reaction, so they attach to the living cell like this, and then you can look at the colors. So this is a later stage zebrafish embryo, 60 to 70 hours post-fertilization. The embryo is alive while they're doing all this. This is kind of it's a zebrafish embryo that's later in its development. It's 60 or 70 hours, and you see all these different colors. Well, these are different dyes that are attached to the dipho-48, and so what they can see, they can kind of track the stages of development of the zebrafish this way, and as the zebrafish is growing, different dyes, colored dyes get incorporated. So what they're doing in this example, in this experiment, is they're first using dipho-647, which is one color dye. They wait a while, then and then they wash it away too. Because it's anything that they add, it'll get attached, right? And that creates like one color here in the development. And then they wait, then they wash away anything that's not attached, because some of it's still not attached. And then they add a different one, a different color. Then they wait, then they wash, and then they add a third one. And what they end up getting are these different colored, the different colors, which show sort of the different stages of the development. And then they visualize this with confocal microscopy. It's just a really super cool thing. I was fascinated when I first saw this, and I thought it'd be cool to show it to you guys. Okay, and then one more thing, early stage zebrafish embryo. So this is 6 or 7 hours after fertilization, and it's, the zebrafish embryo is alive. What they're doing here is it's a much earlier stage than this, and I think what they're doing is they're using a rhodamine, which I believe is just a mitochondrial stain, and that's the red color. And then I think they're using dipho-48, and they're labeling the azides, and that is, I thought it was 48. May have the dye color wrong. But anyway, that's basically creating these kind of images. I think I'm a little more fascinated by this one. It makes a little more sense to me. You know, double check on which dye they're using. But this is a pretty cool example of how this copper-free click chemistry is used in a lot of different things beyond just drug delivery. Okay, back to hydrogel drug delivery. So I was just trying to show this amazing science created by Carolyn Bertozzi's lab, where they use cyclooctane copper-free click reactions, and where those originated. So we're back to this, and this is just showing a completely different use of it to actually generate these sort of cleavable hydrogels. Okay, and so what are some of the follow-up studies they did once they made these? One of the things they did is they injected dye-labeled hydrogel into a rat to monitor the time course of the hydrogel's degradation and in a living organism. So they were able to kind of follow the hydrogel degradation. So here's what it looked like. It's a hydrogel. These are hydrogel microspheres, and what was a microsphere is this sort of injectable slurry of hydrogel particles attached to a linker and fluorescein. And what they're showing, and this is right out of their paper, was they had a rat that had different injection sites, I guess, at different times. They show like 1, 3, 5, 7. There's actually 1, 2, 3, 4, 5, 6, 7, 8, and I think C was a control. The figure legend was that injections of microspheres into the rat, spacing is like 1, 2, up to 8. And each of these is 3/8 the reverse gelation time, which is kind of the, remember, a full RG was like complete degradation of the amount of time to completely degelate the microsphere and completely dissolve it essentially. So they're doing fractions of that to kind of follow, inject, wait a while, inject, wait a while, etc. Okay, and so Rg is the total time for the microspheres to degelate or dissolve. And what this is showing is that these little circles are actually have nothing to do with the drug. They're tattoos. They actually did a little. They opened a mouse tattoo shop where they were tattooing mice, but this showed the injection sites 1, 3, 5, and 7. They're not showing the 2, 4, 6, and 8 on this slide. 1 is the oldest injection, and the 7 is the newest injection. What you can see is that the old injections, there's less of a lump. As you move closer up to like 5 and 7, there's more of a lump. And so the lump is actually the hydrogel. So the gel is kind of in there. And it just shows that over time, the gel degrades. That's an important study when they're trying to do this drug delivery device. Okay? So this is a fundamental study, and it shows that they can watch in real time the degradation of the hydrogel. The key result is the lumps on the skin, which basically means the hydrogel, the injected hydrogel, it decays with time. Okay, and then the other thing they pointed out in the paper is that the degraded or oligomeric PEG, oligomeric just means it's not polymeric anymore. It's kind of partially degraded. The little pieces of PEG, the branched PEG pieces, they're cleared from the injection site. So by the lumps not being there, it shows that not only is it degraded, but it also went away. And if the oligomeric PEG was still around, it would be a big problem, right? Because you don't want all the degraded fragments to kind of hang around. You want them to go away. And then they did 2D ultrasound as just another cool experimental technique to monitor the degradation of the hydrogel microspheres. Same framework, hydrogel microsphere. They have a linker. They're using fluorescein, which is just a fluorescent dye. So it's not really a drug as such, but they're using it as kind of like a potential drug. But that's not the point of this paper. The point of the paper is to just follow the degradation of these microspheres. Okay. Fluorescein, though, is detectable because it's fluorescent, and they can detect it at the site of injection later, for example. Okay, so this is showing they're using ultrasound to kind of follow the degradation of this little hydrogel mass, and you can kind of see it sort of goes from being large at 0 hours, 1700 hours, it's still there, and at 5,880 hours is gone. So that also supports this time dependent degradation. Ultrasound analysis of implants is the figure legend. 2D ultrasound images, so it's a non-invasive technique that allows them to kind of visualize this mass of material. They're using CN1, remember CN? That was the slowest one. That was the one that degraded the slowest. It was the modulator with the slowest degradation. They're using epsilon lysine, which is one of the lysine frameworks we talked about. And it showed these cross sections, I guess. This is like an analytical technique where they're able to measure. It's like a 3D technique. This is a 2D technique. This is a 3D technique that kind of estimates the volume and the size of the gel. And this is a 3D analysis of the ultrasounds using I think a software technique or something, and it shows the decreasing volumes of the implant. And this can be quantified, and you can graph this to sort of show the time-dependent degradation of these hydrospheres. The key result, this analysis shows time-dependent disappearance or dissolving of the hydrogel microspheres. Okay, one more thing that they did that's pretty cool is rather than using PEG hydrogel, what about the idea of just using PEG? Because PEG will kind of immobilize a drug, it's just not as long. And if you do PEG, a linker, a cleavable linker, and then a drug, you can also do the same sort of thing, but this is much simpler because it you don't have to create that whole cross-linked matrix of PEG molecules. All you're using is just PEG, and right. So it's not a branch. I think they're using branched PEG, but they're not. But they're simply attaching the drug via a linker. So without using a hydrogel, they're just using PEG. And the only issue is it's a shorter term drug release. You can't do this for like a year. This might be a week of drug release. So the half-life is about 7 to 10 days. Okay, so ProLynx, there's a different paper. This is another 2019 paper. It's cancer chemotherapy and pharmacology, I think, 2019. So they're using the same drug cleavage technology, but not with a hydrogel in the drug design. They're simply using a cleavable attachment to PEG. So this can be PEG, 40 kilodaltons, cleavable linker, SM38. What's SM38? It's a potent anti-tumor drug. It's actually based on, it's a topoisomerase inhibitor, and this is one of the major drug targets for anti-tumor drugs. It's an analog of camptothecin, which looks like this. And what they do is there's a lot of drug development work around camptothecin and prodrug strategies. Through all of that, people discovered a molecule called SM38, which looks kind of similar. But the one thing is that there's a phenol here on this aromatic ring, which is great because we can attach stuff to it. So this is a great attachment point for a drug delivery strategy. Whereas camptothecin, the original molecule, I think this is a natural product, it didn't have this. It did have a tertiary alcohol, but it's sometimes hard to attach stuff to a tertiary alcohol. Anyway, the phenol is a nice attachment point for drug delivery strategies. All right, so the design of the cleavable drug conjugate, yes, and I was a little unsure about this a second ago, but yeah, they're using PEG, 40 kilodaltons, and this is a branched PEG, so there's 4 of them. So what they do is they have the branched PEG with 4 of these things on it, and it's pretty simple. It's just a modulator, so one of those modulator groups, and then they have a carbamate linkage to SM38, and SM38, basically what happens is once this cleaves through the elimination process, so this one of the protons gets removed and then it cleaves, but then this carbamate linkage falls apart, so it makes CO2. CO2 will be generated from this, and then that also this also then is a cleavable linkage also. So there's a couple cleavage events, and then SM38's kicked out and does the anti-tumor type action. Anyway, so these are a couple of the R groups they use as modulators. They did some optimization, and they found that this one happened to be the best for controlled release of SM38 in a mouse model in terms of having like usable anti-tumor activity. So these ones all have different rates, and this one just happened to work best in a mouse model. But for different species, or maybe different drugs, you would maybe use different ones of these. Okay. All right. So this is just one really cool, real, it's a study that shows this PLX038A as a drug delivery method for a mouse. So what they did, this is a pretty dramatic illustration, day 1, day 12. This is a mouse with a massive tumor. I think it's a xenograft tumor. So they would inject the mouse with tumor cells, and they actually grow, and to a pretty sizable mass. And then they basically at day 1 they inject PLX038A, and at day 12, look at that, the tumor's gone. So this shows a pretty profound example of how this sort of PEG drug delivery methodology attached to SM38 could be used as an anti-tumor drug. and it's a mouse result, and you know all of the human clinical work always originates with a animal like a mouse or rat first. Okay, so these are conclusions about this hydrogel drug delivery stuff. PEG and PEG hydrogels are non-toxic biocompatible drug carrier devices. Beta elimination through E1CB can be used as a linker cleavage mechanism, and the rate of cleavage can be fine-tuned by variation of the modulators. There's a San Francisco-based company called ProLynx. One of my friends works there, and they're doing some cool work with a couple of these drug delivery constructs, like a PEG hydrogel linker drug. It's sort of a slow release methodology. PEG linker drugs is not like a non-hydrogel, but simply just the PEG, the branched PEG. This is more of a quicker release thing. The top one could be maybe used over months or a year. The bottom one is maybe maxed out at about a week or so. But yeah, they're both useful ideas. Different strategies can be used for different therapeutic requirements. So it depends on the drug you're trying to kick out. Okay, so that's the company ProLynx that is doing a lot of this stuff. So you'll probably see more of them as they'll keep publishing papers and pretty cool stuff. The papers that I read were all pretty readable. So if you're interested in this idea, check them out. Okay, so then we're on to antibody drug conjugates, and this one will be next time. All right, so have a good weekend.